Well, good morning, everyone, and welcome to this session. I'm Alice Park. I'm a senior health writer at Time, and it is my privilege to be moderating this discussion with David and Daphne this morning about revolutionizing drug discovery. So both of you have started companies based on pathways that really weren't being optimized in developing drugs, that weren't part of the standard mainstream. So I thought we would start by asking each of you, if someone in drug R&D, let's say a pharma company from a few decades ago, were to come visit your labs today, what would they see that they would recognize and find familiar, and what would be different, or would everything be different? Maybe, David, we can start with you.
Sure. Well, so happy to be with both of you guys today. I think that scientist would immediately recognize that, oh, wow, you guys are trying to find new uses for the medicines that you're working on. So at Every Cure, we repurpose medicine. So that's something the drug companies have been doing forever. Thinking about, it can work for this disease, what other diseases might it work for? But I think they'd be really surprised by a couple of things. First, they'd be surprised that we were looking at that post-patent exclusivity, for generic drugs where it's actually not profitable to find a new use for the medicine, so that would be shocking. And the second would be that we're utilizing artificial intelligence to try to make these matches, every drug versus every disease. What is this thing that is, trying to help find a new use for these medicines? But I think that they would be really excited about it. I think that, everyone in the biomedical industry, we want to help patients with the drugs that we have or the new drugs that we develop. The system just hasn't been created to make sure that medicines are used for all the diseases they can treat.
Okay, Daphne?
So a few decades ago, drug discovery was very much a matter of trial and error. You kind of hoped that you had an insight or that you did some experiment that gave you some insight. And what we're doing in our work, similar to David, I think everyone would recognize the endeavor of trying to discover medicines, but our discoveries are anchored firmly in human genetics. And a few decades ago, the human genome didn't exist, right? This is a relatively new development as these things go in drug discovery. And the other side of it, which I think is maybe an even more direct answer to the question that you asked of what if somebody were to come visit they would find the most surprising, is the extent to which we juxtapose that human genetics with the generation of massive amounts of cellular data that you couldn't even measure in human cells multiple decades ago. And so we have these lab robots that perform massive experiments with microscopes and sequencers and entire workflows where a robot basically works autonomously for weeks without a cell ever being touched by human hands, all of which is controlled by lab automation powered by AI, and then all of that, to David's point, gets filtered through an AI lens that often discovers the starting point for the insight that, of course, is then vetted with and developed with a scientist, but the original insight comes from the AI being able to take this massive amount of genomic clinical data and cellular data and putting it together in a way that the human mind simply could not ever do.
We're going to get back to that data, but so how much of it would be the traditional so-called wet lab, the lab benches that we're familiar with from, old photos of scientists, doing their experiments with beakers and all of that. How much of that is still a critical part of the development, and how much of it goes on in the black box of the computer?
So speaking for us, our R&D function is almost exactly 50-50 people who work with life sciences and people who work with computational sciences, with the lab automation people being kind of a little bit in the middle. And there are still some traditional lab benches where people do some small-scale experiments, kind of assay development, things that are not at scale. But then if you look at it from the perspective of how much of our lab space is automated labs versus how much of it is more traditional labs, I would say that is approximately 50-50. The computational part is that doesn't include the computational part, which is the other 50% altogether. So really, traditional lab experiments are, I would say, less than a quarter of our footprint and a very small fraction of the data that is being produced.
Okay, interesting. Now, I want to have a quick show of hands here, make sure you're all awake. How many of you are familiar with David's journey to creating Evercure? Oh, good. Okay, we've got quite a
Got some friends over there. So
Yeah, all right.
It's a biased sample.
All right, so the friends don't count.
friends
But it
count.
looks like there are some people who aren't familiar. So tell us briefly about your personal journey and why you created Evercure.
Sure.
Evercure, sorry.
Sure, yeah. So I went from being a healthy third-year medical student at the University of Pennsylvania. I wanted to become an oncologist. My mom had died from cancer when I was in college, and I was well on my way to take care of patients in her memory when I got really sick from a horrible disease called Castleman disease. I spent about 6 months in the ICU, nearly died on 3 occasions, had my last rites read to me, said goodbye to my family, and I got a lot of chemotherapy that saved my life, but I kept relapsing. A total of 5 times, almost died from this disease. And after the 4th time I almost died, when my doctors explained that we were out of options and that I was going to die very shortly because I was approaching the lifetime maximum of the chemotherapies they were giving me, I thought about the fact that those chemotherapies they were giving me weren't made for Castleman disease. And so I was like, wait a minute, you're telling me we're out of options, but you've just saved my life a few times with drugs that were made for other diseases. How do you know there's not another drug made for another disease that could also save me? And have we actually tried all 4,000 drugs for Castleman disease? And of course, we hadn't. And so I went on this mission as a third-year med student to try to figure out, is there an existing drug that could save me? And I did a series of experiments on my blood and also lymph node tissue. And I discovered that a communication line in the immune system called mTOR was in overdrive, and decided to try an mTOR inhibitor for the first time ever, an mTOR inhibitor for Castleman's, and it had been approved on the market for decades.
But for other diseases.
For other diseases, and it had never been used before for Castleman's, and that drug worked, and it's now been over 12 years that I've been in remission, approaching 12 and a half years on July 5th, and the moment that drug started saving my life, I have not been able to stop thinking, how many more drugs are sitting at our local CVS or Walgreens that could save patients today that are just waiting there to be uncovered, that no one is doing the work to systematically uncover. And so that's been my mission. Now that I'm so lucky to be alive, thanks to this existing drug, how can we unlock more of these existing drugs to treat patients who are suffering today?
So in the industry, it's called repurposing. Yep. And as you said, it's long been a part of the pharmaceutical industry, but not a very important part, because it's usually it's drugs that, as you said, have gone off patent, they're generic, they're not the money makers anymore, and they sort of put them aside as they look for the next big thing, right? How important has Evercure been in sort of revitalizing the importance of repurposed drugs and just refocusing the attention on them, like you said, and their potential, which clearly there is quite a bit of potential there?
Yeah, it's such an incredible opportunity. I mean, every patient that we save with a repurposed drug, and we think about, oh my gosh, this drug was there and they wouldn't be alive today if not for it, it means the world to us. We've now repurposed a total of 14 treatments over the last decade. We've got 12 active programs right now at Evercure that have the potential to transform many, many more lives. I think that what's so interesting as we've gone on this journey the last few years is speaking to folks who work in drug development and learning that the average, the typical drug in development will have between 15 and 25 diseases that are considered for that drug over the course of its R&D. And really tough decisions have to be made. Do I pursue this disease or that disease? And a lot of times it's not because the biology isn't strong. It's not that that drug won't work. It's that it's hard to make a business case for that to be the first disease to pursue. And so companies have to make really tough decisions to put additional opportunities to the side. And of course, they can only pursue a few, and then that drug becomes generic, and all research and development ceases. Not even when it becomes generic. When it gets close to becoming generic, that's when R&D will stop. And so there's this proof that we know that companies consider a lot of conditions per drug. We also know that drugs are often approved for multiple diseases because companies do the work to prove that out. And we know that there's so many more opportunities. Drugs don't stop having additional potential just because they're generic. I mean, if anything, at that stage, we know more about how the drug works. We know the safety profile of the drug. The drug might already be being used off-label by doctors. We might actually be getting clinical data showing it can work in a new way, yet that's where our system stops. And so at Evercure, we want to make sure that these amazing drugs reach all the patients possible.
Can I just add something to this? Because I think David and I were having a fascinating conversation
Yes.
before we started, which is there is, I think, a major fallacy in thinking about the relationship between a drug and an indication as the primary relationship, because a drug doesn't cure a disease. A drug intervenes in a specific biology, and diseases are a dysfunction in one or more biologies. And if we were to sort of move away from the disease to drug mapping.
But that's been the standard, right,
It
for
has
how
been.
drugs are developed. It's how the pharmaceutical company is built, because you can't just say, we're going to pursue this thing, let's see where it works, right?
And I think it's partly on the drug companies, and I think it's partly on the regulatory approval process, which has to have an indication. That's
It's
what it's
going
called,
to
an
do this
indication, the
in
thing that
these
is on the label. And so but really that is not how drugs work. Drugs intervene in a core underlying biology that is disrupted in the disease. Most diseases involve the disruption of actually more than one biology. And so if we were to instead change our taxonomy to a mapping between diseases and biologies and a mapping between biologies and drugs, we would be able to not only do repurposing so much better in the way that David has already started to do, but also think about the right combination therapies and which drugs are interchangeable for which other. And I think that would kind of transform how we do drug development and drug delivery. Whereas I think right now what we're using is a taxonomy of disease that is frankly incredibly antiquated. It's based on a very coarse symptomology that is based on very coarse level observations that a physician in the clinic can glean quickly. And that is just the wrong way to think about disease.
Right. And it's a very limiting approach, right?
Yeah, I agree. And I was going to just follow up to Daphne's point. We were talking about a rare condition called Bachman-Bupp syndrome. It's named Bachman-Bupp because of two amazing doctors and scientists, Dr. Bach and Dr. Bupp, who discovered that children with this really devastating neurodevelopmental condition and also inability to move and function have a mutation in a gene called ODC1. And what's so interesting is that decades ago, a drug called DFMO was made to treat African sleeping sickness, and the way that it would work to treat African sleeping sickness is to inhibit ODC1. Well, we can utilize this old African sleeping sickness medicine for Bachman-Bupp syndrome because the exact same therapeutic target is shared between African sleeping sickness and Bachman-Bupp syndrome. And so rather than considering it Bachman-Bupp syndrome constellation of symptoms, it's Bachman-Bupp syndrome, this ODC1 gain of function mutation that we can treat with this existing medicine. And what we've seen has been transformative in the 6 patients that have been treated. They no longer have this accumulation of polyamines. One of my favorite things that was put on a slide by our chief scientific officer, we were talking about the improvement for these 6 kids when they've been treated with DFMO. And one of the things he wrote was HUGS, H-U-G-S, in capital letters. And I immediately thought, is that an acronym for something? And he said, no, David, he hugged his parents for the first time ever. Like, hugged his parents for the first time ever. Like, that's an endpoint that we need in clinical trials, right? And that's so meaningful, using this old African sleeping sickness drug to enable things that these children and these parents never imagined.
And this is part of, Daphne, you've described what you do as developing drugs entirely conceptually. Explain to us a little more what you mean by that.
So it's not entirely conceptually, because we absolutely do a lot of lab experiments.
Of
And
course.
I'm not one of the people who believe that there is a magic wand that in the computer
Right.
will say, aha, this is a drug, and then you're done, and it's already in
But
the
in
clinic.
the designing.
But yeah, and so what we really do is follow the exact approach that I just articulated, which is try and go into high content data where you actually measure the underlying biological phenomena that happen in the context of disease. And we now see those manifestations in human clinical data that can be collected and often is collected very readily from patients, whether it's imaging, whether it's omics from blood and tissue, different types of blood measurements. And you can really see how the different biologies there are being disrupted in the context of the disease. And then you can take that and say, okay, how do you translate that into the biological mechanism that we need to now intervene in order to make a drug? And so you map what is a very broad category of a person has this type of disease like ALS into what are the specific biologies that are disrupted. You create a lab surrogate for those biologies, and you're very quickly able to sort of align what you see on the cellular side with the human clinical mechanism, and that gives you your intervention target anchored in human genetics. And then, of course, you obviously need to make that into a drug, and so we leverage the best of breed of approaches, both from ourselves and from others, to very quickly identify a compound that one can use in order to make that intervention in that specific biology, whether that is a small molecule or a biologic or an RNA therapeutic, which of course is a wonderful category, something you can develop really quickly these days, and so the conceptual part comes in creating that map of the disease biology, understanding what is disrupted and how one intervenes in that by in the right biological and cellular mechanisms.
So this whole idea of not seeing diseases as silos, but really looking more at the biology.
Yes.
Where does In Citro, your company, where does that data come from? Are you getting patient samples? Are you collaborating with academic centers? Where does that data come from?
So we have two kinds of data, and we bring them together in a very unique way. One of those is human clinical data, which we get from a combination of biobanks and academic medical centers. So biobanks would be things like the UK Biobank in the UK, all of us in the US, as well as more specific, specialized cohorts that come from academic medical centers, where we can really get a deeper understanding of the specific disease biology. But that's great, and human data is an incredible source where you can understand the causality of disease by aligning human genetics and human clinical outcome, which is very hard to do any other way, because you cannot do experiments in human beings in anything other than a clinical trial, and human genetics is effectively an experiment of nature that was done on, all of us, right? But how do you then do the actual experiments where you could say, okay, I'm going to try and intervene here or there or there? And for that, we basically print billions and billions of cells, different types of cells that really correspond to the right human biology. These are not some random cancer lines. These are, for example, motor neurons that are induced pluripotent stem cells, and we intervene in them, measure them with exquisite precision and scale to say, aha, if we intervene at this gene, it reverts the biology that we saw on the human side. And that convergence, that triangulation from the human clinical data and the cellular data that we print in-house, like I said, I think we're 22 billion cells and counting that we've printed and phenotyped, is where we find those intervention points.
David, these pathways that you're talking about, that you and Daphne talk about, that are common to a lot of different diseases, and even the mTOR that was identified in your Castleman, which we now, I think, know a lot more about, right? That mTOR pathway today than we probably even did when you were treated. Was what you are doing in terms of repurposing and finding those commonalities possible 5 years ago, 10 years ago?
Yeah, it's really just in the last 3 to 5 years that what we're doing has become possible. We were really fortunate to receive funding from ARPA-H about 2 years ago to build out an AI platform where, as Daphne mentioned, there's so much that's critical to be done in the lab to understand human biology and disease biology. At Every Cure, we're actually able to leverage all of the scientific experiments being done all over the world. We use existing medicines. We don't need to come up with a new drug, and we can rely on the experiments that are being done constantly all over the world, the publications from those, and our powerful AI platform essentially is looking across all of the world's biomedical knowledge in an organized, we utilize biomedical knowledge graphs oftentimes, but organize the data and then utilize machine learning models to look across all of human biology. We train them on known treatments, and then we ask the models to find and to score from 0 to 1 how likely every drug is to treat every disease. When we ran this model for the first time 3 years ago, and it's, as far as I'm aware of, the first time anyone had ever done this, it took us 100 days to get the results back. It's a total of 75 million calculations between, you get a result from 0 to 1, and it's a normalized or relative score. So a 0.99 for lidocaine for breast cancer is better than a 0.95 of Botox for major depressive disorder. Imagine every drug versus every disease, but by ranking them and creating a normalized score, then our humans can go to the top of the list and say, among all the drugs out there and all the diseases out there, what are the best opportunities to help people? They may not be people with a disease that you've ever heard of, but it's people with a disease, and it's people where there's a drug, where there's a nice, strong biological connection to that disease, and then we want to go there. And so, like I said, it took 100 days to generate those scores 3 years ago. Now it takes about 17 hours, and the predictions are getting better and better and better. We find so many interesting and amazing opportunities to use medicines in new ways. And so it's not that we're not using laboratory-based data. In fact, we're actually utilizing all the laboratory data that everyone's generating all around the world to feed our artificial intelligence model to find matches, and then we go do further laboratory work to confirm those things. Then we do further clinical trials to confirm that those initial insights and hypotheses we have are actually going to reach people. And then the last thing I'll say is that making a prediction is incredible. Doing the laboratory and the clinical trial work is critical, but we also take responsibility for that last step. After you've proven the medicine works, how do we actually get it to people? How do we work with drug companies to make sure there's sufficient supply, despite the fact these drugs are often generic? How do we work with guidelines committees to update guidelines, regulators? But it's about making sure that we're not just making predictions or just doing one part of the process. It's about being end-to-end, because these old drugs will fall through the cracks if there's no one to champion them getting from discovery all the way to people.
I want to go back to this supercharged master list that you can generate in 17 hours of every available drug that's now approved
Yep.
and every disease,
Yep.
right? Are you getting bombarded by biotechs who are asking for access to this list, and who has access to it?
Yeah, so biotech companies are very interested in us applying this approach to drugs that are not yet approved, or drugs that, and in particular, drugs that are on shelves within drug companies. So we've stayed really true to our mission. We're really focused on FDA-approved drugs for diseases they can potentially treat. So we haven't yet partnered out beyond approved drugs. Right now, this list, which ends up being millions and millions of rows long, it's just a drug disease match with a score next to it. And for us, it's a great starting place for our medical team. Our med team reviews hundreds, and in some months, up to 1,000 of the top repurposing ideas that come from our platform every month. Collectively, in the last year and a half, we've reviewed about 13,000 of the best repurposing ideas from our AI platform, humans reviewing 13,000, and finding so many incredible opportunities in there. And so we're excited to continue to share this list as a starting place for academic researchers and medical scientists. But for right now, we're staying really focused on existing medicines for diseases.
That score seems like it'd be the holy grail of, where to start, right?
It's a great place to start. And I think that's really important, is that, a lot of people hear that Evercure is putting so many resources into this, and we've got an amazing team. We've got such a great tech team and medical team. And even still, the top hits on the list, I wouldn't be ready to say, you should take this medicine because it's scored #1 by our AI platform. Scoring #1 in our AI platform means that our humans should look at it, and we should consider whether clinical trials are needed, whether laboratory work's needed, right now. But we shouldn't be, using the list to go right to patients. But it's a great starting place, and I would much rather start there.
17 hours versus potentially years, right? Of postgraduate work and decades.
If we tried to get humans to try to figure out a rank-ordered list of every drug versus every disease, probably decades of time, and then you got one list. And so even if it's not perfect, it's an incredible starting place, and it helps our brilliant humans to figure out, where do we start?
So Daphne, that's a really wonderful example of the power of AI and being able to take all the data that we have now. And you're known, obviously, for thinking ahead about how we can use this really powerful technology to address needs. So tell us about some of the targets that you've been going after, and how AI models have really telescoped the process of getting there.
So I will give you 2 examples that we're particularly excited about. The first of those is a program that we are within line of sight for a development candidate for a disease that I think many of you are familiar with, which is ALS. ALS is one of the most horrible diseases that you can get. When you talk to somebody, or like when you talk to a neurologist, every patient who comes to me is terrified that they have ALS, and frankly, no matter which diagnosis I give them, they are relieved that it's not ALS because it is such a horrible disease. It basically kills you within 3 to 5 years, and as a community, as of now, we have basically offered nothing to the vast majority of ALS patients. They die within 3 to 5 years. And what we were able to do is, coming back to your question about which data we've generated, we printed 12 billion motor neurons. We took 6 billion of those were phenotyped using high-content super resolution microscopy in order to identify what happens to a motor neuron as it gets ALS and dies. And so you can actually, and the AI found what we call a disease axis. This is what it looks like. This is what ALS looks like at the cellular level. We then took 20,000 genes and tried to see which of those reverted that ALS phenotype. And that allowed us to identify novel revertants of this disease. Now, in the same way that David said, well, I wouldn't take the top hit from my machine learning algorithm or AI algorithm and just put it into the clinic, we then took those top hits and vetted them using a whole score of other kind of more fine-grained assays, say, this is a known consequence of ALS. Does it revert that? Does it revert this? Does it revert that? And we were really impressed that the top hits actually reverted multiple consequences of ALS, not least of which is a functional consequence, which is that damaged neurites in ALS neurons do not grow back, and they do grow back in normal, healthy individuals, and we reverted that neurite outgrowth deficit. And so that gave us the targets, which we're now turning into drugs. So that's one example of an insight that came through our platform. Unlike David's approach, we don't screen for existing drugs. We screen at the genetic levels. We screen for a gene, which would then be a target against which we would then make a new drug.
But you're able to do that because you can see that process now in its entirety, which we have not been able to do outside the body before.
And that is a combination of measurement, sophisticated lab technologies that we've put in place to create those high-content measurements, and the AI that allows us to see things in these cells that a person cannot see. You show these cells to a human being, their eyes glaze over after the 15th or 20th image that you show them, because they all kind of look the same. And they certainly can't put a language to why do these cells look a little bit different than those cells. But an AI can really see that. So that's one example, and I'll give you a different example. I'm giving you two specifically because they are so complementary in how they've done the work. The second one that we're really excited about is we used AI, in this case, on clinical data to, for the very first time, measure brown adipose tissue. I know longevity is a very big theme at this conference.
Is anyone here who knows what brown adipose tissue is? Okay.
Wow, that's a lot.
It is.
So for those of you who didn't raise your hands, brown adipose tissue is brown fat. Babies have brown fat. Brown fat is a metabolically active fat that actually burns calories rather than stores calories as fat. And so it's the more of it you have, has been shown in animals, the healthier you are.
It's actually fat you kind of want, I guess.
Fat you kind of want, but unfortunately you lose it as you age. It's one of the consequences of aging. And so people have studied brown fat in mice for probably about 15, 20 years. And the mouse genetics that increase the amount of brown fat and make the mice healthier haven't translated to humans, because you know what? People are not mice. I know that's really surprising to a lot of people, but they're not. We were able, using AI for the very first time, to quantitate the amount of brown fat that people have, and actually have a quantitative measure saying this person has more, this person has less, something that a human can never see by looking at, say, an MRI image. And by doing that, we were able to, for the very first time, identify the genetics of having more or less brown fat, which is maybe a bit of an explanation as to why some people can eat as much as they want and they never get fat. My husband is one of those. And other people, they as much as look at a donut and, they gain 15 pounds. And so what we were able to do is get at the genetics of that, and then we took the first of those genes that came off of that list, and we put that into a mouse, and sure enough, the mouse developed a lot of the mouse lost a lot of weight, and unlike the other weight loss drugs, it was entirely fat mass. None of that was lean mass. And what we're seeing is basically a process in which white fat turns into brown fat. So it's actually taking the bad fat and turning it into good fat. And so we're really excited.
Here first, the next GLP-1.
And the reason I'm highlighting that is because both of these are examples of collecting lots of data. In one case, it's in cells. In the other case, it's in humans. In both cases, it's drawing in insights from the other side. So from the cellular side, on the brown adipose tissue, and in the ALS example, we drew an insight from the human side that allowed us to triangulate on a high confidence target, which we then selected and then are developing into a drug.
Okay. Before I go on to my next question, just want to remind everyone we will have Q&A. Daphne and David have agreed to answer any questions you might have, so keep them in mind, and we'll come back to that toward the end. So, David, I wanted to ask you, what is that relationship and interaction like with pharmaceutical companies when you're taking something that they've essentially put on the shelf? What are they asking for in return, or what's that interaction like?
Yeah, great question. We work with companies once we found a promising repurposing opportunity in a few ways. One is to see if they'll donate the drug to us for clinical trials. Drugs are expensive to include in trials, even when they're generic, and so that's really important. So one is donating drug for the trial. Another is providing insights about the drug. No one knows these drugs better than the folks who develop those drugs, and so we want to work with these companies to understand how they work, observations they've made, whether they're, if the drug worked really well in this way or maybe it didn't work well in that way. And so it's after we've made the repurposing connection. So it's one of our active programs. That's when we work with drug companies. And then we recognize that so out of our 12 programs, only one of them is a drug that's currently on patent. And that's not too surprising because 80% of all drugs are generic. And so it's maybe not too surprising that about 90-something% of our portfolio is a generic drug. And also generic drugs have more data known about them. So that's another reason why our platform might be surfacing them at a higher rate. So even after we get a drug donated for trial and we can do a trial, we still take responsibility for making sure the medicine gets to patients. We recognize these drugs are off patent. The drug company is not going to put together a sales force for a drug that's generic, right? So we work with them to understand how they've marketed the drug, what they know about the drug, and then we take the responsibility for making sure that the medicine actually reaches patients. And I might just share one example that may be tangentially related, and that's around a rare condition called DATA2 syndrome, where kids have a mutation in a gene called CECR1, and as a result of it, they accumulate dozens and dozens of strokes, and they die in their teenage years. But about 20 years ago, a researcher, or actually a physician, had a patient with DATA2 and a patient with a form of vasculitis, and had just tried given a TNF inhibitor to the patient with vasculitis, and thought maybe I'll try the TNF inhibitor in the patient with DATA2, and that kid stopped having strokes. He treated a couple other patients like that, but it was about 15 years between that first patient not having strokes and actually getting guidelines together so that other kids would get a TNF inhibitor and stop having strokes. In that intervening time of 10 to 15 years, hundreds of kids died from DATA2 syndrome from having strokes when the world knew there was a TNF inhibitor that could help them. So it's not just that we have to discover these things, we have to compress the time from discovering that a drug can be useful in a new way to proving it. So there was actually a really important retrospective study that was done that showed kids just stop having strokes after they got TNF inhibitors. We got to get retrospective data rapidly, and then we got to start getting the word out as quickly as possible, because to me, that's the ultimate tragedy, right? When the world knows, we know a drug could stop strokes and stop these kids from dying, yet the world, we haven't done enough to get the word out, to do the right work rapidly, so that way other kids don't suffer from these things. And so I share that story just to say we love finding things, we love proving they work, but we got to make sure they reach all the way to patients.
And in terms of that testing process, as you mentioned, because these are repurposed drugs, there's already been safety efficacy data for certain indications all the way through the FDA, which is, has its own stringent rules. Does that make it any easier for the clinical trial process in looking at a different indication?
Absolutely, yeah. Once the drug's already on the market, if you're using the drug at the same dose and similar dose and formulation to what it was approved for, the FDA will oftentimes grant an IND exemption, which makes the clinical trial process substantially less expensive than if you're doing it under an IND. And what we can set our goalpost as is what is the endpoint and what is the trial that will help a doctor to know whether this drug works or doesn't work. And it's not based on regulatory precedent or what's going to get the FDA label change. It's just what is the right trial for a doctor to be able to make an informed decision about whether it works or doesn't work. So we can work with doctors, we can work with patients, we can design trials in a way that's really about, we haven't included hugs in our endpoint yet, but it's that sort of thing. The things that are really meaningful, and by being able to do trials in an efficient way and go after the really meaningful things, then that allows us to just do more trials with the funding we have.
Daphne, in terms of the way you're using AI modeling in particular, right, in the design process and in even potentially identifying targets, how is that affecting when you go to the FDA, or how do you anticipate, or have you started to have conversations about this sort of new era of drugs where they might not have been discovered by serendipity or by medicinal chemistry the way they have for decades, but by this algorithm? Have you started having discussions, or how do you anticipate working with the regulatory agencies on that?
Yeah, so we have just begun our first interaction with regulatory agencies for our first drug, which is going to be in the clinic, if all goes well. I guess this is not wood, by the fall of this year. I think there are two answers to that question. There's the conservative answer, and I would say the more ambitious answer. The conservative answer is that, frankly, the regulators don't actually care how you discovered the drug. They care about how what preclinical evidence you provide.
So they're just looking at the endpoint. Like, did you meet the target?
They're looking at whatever animal data that you have, animal safety data, some efficacy data, which is less relevant perhaps when you go into phase 1, but they don't really care if the drug came out of the brain of a scientist, of some reading a paper in the literature, or because of an AI algorithm. So that's the conservative answer. I think there is a more ambitious answer, which I would really hope that we can, over time, move regulators closer and closer to, which comes back to some of the conversations that we were having earlier, which is we are now, because of the tools that we have available to us, both the measurement tools on the one hand and the ability of AI to interrogate those tools, being able to create a much more, I would say, precise and compelling map of the biological function of a drug and the biological map of a disease. And if we were to have that not only be part of the initial package of what it takes to get the drug into the clinic, which is frankly oftentimes a fairly low bar because you just need to ensure safety, which is great. I mean, it's not a low bar, it's a very important bar. But at the end of the game, when a drug is approved, you want the regulatory endpoints to match the underlying biology versus some, potentially self-reported, qualitative, highly variable outcomes that are currently being used for the approval process. So for example, if we're able to show in our ALS drug, which is now starting to happen, that the molecular biomarkers that correspond to neuronal death are being affected by a drug, that is an endpoint that should be actually not a secondary, but a primary endpoint that is being used as part of the approval process. If you can look at things that are much more objective measurements of patient health, not qualitative questionnaires like, how are you feeling this month? Are you feeling better than you did last month? Which is frankly...
Or things like the hugs, right?
Yes,
which is a very
exactly.
physical
which are great, but they're also oftentimes very qualitative and rely on patient recollection. But if you can use wearables, for example, to actually measure whether people can lift their arms better, whether they're breathing better in the context of ALS, which kills you by eventually shutting down your respiratory system, those are much, much better endpoints. They're more rigorous, they're more quantitative, they're not subjective, they're continuously measured, and I think we should move more and more towards endpoints that are grounded in disease biology and with a kind of quantification that only AI can provide you.
This is part of the discussion, I think, that's been going on for years now on surrogate endpoints. And as we get smarter and smarter in understanding the biology, like which ones really indicate
100%.
whether we're having an effect on a disease and the symptoms of a disease.
And I think for those endpoints, you're right. Oftentimes we don't know a priori which ones are going to actually move the needle in terms of patient outcomes. But I will point to NFL, which is neurofilament light, which is the endpoint on which the ALS drugs some of them were speculatively approved, and we're now seeing dramatic improvement in a small subset of ALS patients, the one with the SOD1 mutation, where you actually see disease reversion. You actually see patients getting better, not just the slowing or stopping the progression, but some patients actually are getting better. They are moving better, which is amazing. So sometimes you just have to take a little bit of a leap of faith. And if you we were privileged a few years ago to have an amazing conversation with Sue Desmond-Hellman, who was an amazing physician scientist who led some of the early developments of HIV drugs. And she said very clearly that if we had not had viral load as a surrogate endpoint, as opposed to, HIV outcomes like getting cancer and dying, we would never have the HIV drugs that we have today, because the development cycle would have been years rather than weeks. And increasing the velocity of clinical development is the most effective lever that we have at our disposal to get more drugs to patients.
Yeah. And speaking of increasing that speed, just really quick, I want to ask you, Daphne, your thoughts on, even if we're at this point, right? Like, as you said, the regulatory agencies are just looking at the final, at the output data. Do you see in the very near future us being able to look at AI-based models, disease in a dish type things, or simulated models of how X drug might affect X process in a disease, and then be presenting that to regulatory agencies?
I would certainly hope that should be part of the evidence base for both efficacy and safety. I am not convinced that animal models in some cases are actually more accurate at predicting what will happen in a human relative to some of these in vitro models, although I would say that there are some things for which we currently at least do not have a surrogate for what happens in a system. And people are working on organs on chips and things like that, but those do not yet recapitulate the complexity of how a drug is metabolized and travels to different parts of the anatomy of a full organism. So I think we can certainly, and not only could and should make steps towards using these more scalable and more humane models in order to reduce the reliance of animals. We do not yet have, I think, the silver bullet that will allow us to completely do everything in a dish and not do some of the preclinical work in animals. I wish there was, but.
Yeah. Before we go to the audience, I wanted to ask both of you the same question. And so we started by asking you, how your labs look different from those 20 years ago. If we look 10 years in the future, what do you expect to see or hope to see in drug development, and how do you think drugs will be developed and tested and approved? David, start with you.
Sure. Well, I hope that our AI platform continues to get better and better, where our humans are reviewing a handful of the best repurposing ideas, and those are the handful that we're moving forward. And maybe in 5 or 10 years, we feel even more confident that an AI prediction maybe could go right into a patient. I should even share a caveat that 3 years ago, when we ran the algorithm for the first time, we had a patient who was dying in Vancouver from Castleman's, the disease that I have. And so we actually did give him the top scoring drug from the algorithm because he was out of options. We had no idea what to try for Alan. And it worked. And Alan's alive 3.5 years later. So in some rare circumstances, I think it's reasonable today in 2026, or I guess back then in 2023, to take these sort of long shot approaches using something from AI. But hopefully in 5 or 10 years, it's going to get better and better, where maybe we feel more confident than I feel right now in going right to this is scoring really well. Let's try it in patients. Of course, that's only of approved medicines. I wouldn't want to do that with a drug where we don't know the safety profile that's on the market. But within approved medicines, I hope that we're there.
But I want to reiterate that even that long shot that you said was based on your score, on every cure score, was a much better shot than the ones you had, right? It was based on a lot more data than
Yeah, then like the
chance that
sirolimus
you
for
had.
me.
Right.
Yeah, it's a great point. Yeah, I think I think you're right.
So already, there's progress there.
That's a great point. Yeah.
Okay. Daphne?
So I completely agree with David. I think we are seeing a massive acceleration of our ability to discover high confidence hypotheses on what intervention will address a particular disease biology, a particular disease. So to parallel David's, what was it, 117 days to
100 days to
100 days,
17
17 hours. We had a similar acceleration in our work where we started out, our first program took 4 years because we were building the platform
Yeah.
at the same time. The brown adipose tissue program that I mentioned earlier was less than 9 months from the time that we first said, let's even see if you can see brown adipose tissue, to the time that we had a tool compound that showed efficacy in an animal. So acceleration on the discovery side is absolutely possible, both for the discovery of a biological hypothesis and for taking that hypothesis and turning it into a compound in cases where we're not doing repurposing and we actually need to make a new drug. And the bottleneck that we really need to resolve at this point is actually clinical development. How do we compress the timelines for for all of the steps, ranging from the time that you first submit your approval to the regulatory agencies, to the time that you are able to recruit patients, measure the outcomes in those patients, and then at the end of the day end up with a regulatory package that you submit for approval. I want to make it clear, this is not a silver bullet. Certain timelines cannot be compressed. If the disease takes a certain amount of time to progress and you need to show that you're addressing that progression, that's the rate at which biology moves. Doesn't matter how many GPUs you have, you're not going to accelerate that. But there are a lot of parts of that process that are incredibly inefficient, incredibly slow, and that can be accelerated if we put our minds to it. And I would really, really hope that in the next 10 years, we have people who can see that and help us considerably compress those timelines so we can test things faster and take some of those incredible accelerations that we're seeing in the early stages and much more rapidly deliver them to patients.
Right, and help more patients.
Yeah.
Okay. Wow, lots of questions here. All right, there's a mic going around, so let's see here in the front.
Thank you. Thank you so much for this. I'm Elizabeth Cohen. I'm a professor of medicine at the City University of New York, and I chair the science committee for the All of Us research program, which is 880,000 people across the nation, enriched for populations that haven't benefited from research in the past, rural populations, women. Thank you for using our data. How can we help? We want to tee up the scientific program 2.0, so we would love to tailor it to do this work.
Francis and I were talking about this literally over dinner last night, so thank you for asking that question. Yeah, wonderful question. So glad you asked that, and thank you for your important work. I think that there is a huge opportunity with all of us, and thank you for your work on all of us, to up-level it by taking the incredible work that was done exactly as you articulated on diversity, which is so much better than what we have had in UK Biobank, but taking some of the benefits that we've seen in the UK Biobank and incorporating them into all of us. Specifically, there's so many measurements that we're collecting from those patients that are not incorporated into the All of Us corpus, and that I know, based on personal experience in our work, give you a tremendous insight into the disease biology. So the more of those measurements that you can incorporate, whether by a concerted phenotyping effort, as was done in UK Biobank, or simply taking what is in the medical records, not just the clinician notes, not just the ICD codes, but the actual data, images, blood tests, and so on, and making that available to researchers, that would supercharge our work. So please.
We're there, yeah, we'll connect.
Yeah.
Okay, let's go to this side. Yes, here.
This is a two-part question.
A mic is coming. No, that's fine. We just want to hear you.
The question is, if a family has a mutation or a rare disease, how do they query every cure? The second is that there is a drug that in cultures of neurons will increase neurite outgrowth, and in mice will increase the little projections on neurites that make synapses. It's a generic approved drug, how can we look in your system to see if we could give that to someone with ALS? Because anybody who knows someone with that disease would do anything.
Yeah.
David, start.
Yeah, so for patients and families that have rare diseases and they reach out to us, we are currently not positioned to be able to say, here are the list of drugs for your disease. In part because, like we said, we don't have the confidence that the number one drug is necessarily the drug. And for some rare diseases, there isn't actually a repurposed drug that's going to be out there for them. We do point families towards a really great machine learning model that we utilize called TXGNN, txgnn.org, so they can actually type in the disease and get a rank ordered list. So it's like what Every Cure does, but it's actually one of the tools we use. But I think that the second point you made around a generic drug that you think could be helpful in ALS patients, we want to hear about those ideas. We love when our AI platform predicts promising things. We also love when people come to us and say, this helped my child, or we have this insight from the lab. There's literally hundreds of repurposing ideas, maybe thousands that are waiting to be uncovered. So maybe you can share that with us after this.
Based on understanding the biology.
Exactly.
Yeah.
So
Okay.
should I answer the second question?
Sure.
So I would need to know more about the target for that drug to see whether it makes sense in the context of ALS. I would say that there is several ways in which one can increase neurite outgrowth. Some of those target the underlying biology of ALS, others do not. Our drug not only does that, it also reverts the many biochemical markers that exist in ALS, which are the underlying root cause for the neurite outgrowth deficit. So whether this drug actually goes after the right pathways or not is something we can discuss offline.
Okay. And unfortunately, we only have time for one last question, so let's go to this side over there.
Thank you. How do you see the relationship between humans and machines evolving in your labs and your workflows over the coming 5 to 10 years?
Okay. Humans and machines in our labs work incredibly closely together, and I'll give you one example of a recent use of agentic AI for assay development. Assay development used to be this thing where a person would say, oh, I want to see this kind of signal in cells. It would take 9 months for them to try this. Oh, we're not quite seeing the right robustness. We're not quite seeing the right separation. We now have them working in tandem with both agents, agentic AI on the one hand, and lab automation on the other, to sort of not only propose the right experiments, design the right sort of set of interventions, mapping the plates, running them through the lab automation, and then the human says, yes, I think we're seeing the right signal or not. We're shrinking that time from 9 months to a small number of weeks, where the rate limiting factor, similar to in clinical trials, is how quickly does the biology actually move. And so you can compress timelines by the symbiosis between humans and machines on every single step of the process, which is how we've gone down from like 4 years to less than a year, is by consistent kaizen of compression, where the machines just supercharge the humans. Think of it as like a machine exoskeleton for the humans.
Okay. David, Daphne, thank you so much. We're clearly at a very important transformative period in drug discovery, so it's all good news for patients. Thank you.
Thanks so much, Alice. Thanks, guys.